So hello students, all of you, welcome back to your own channel Vedantu NEET English. This is Basavaraj Sir, your biology master students. I hope all of you are doing great, amazing and not sleepy. Okay. Now students, now this is a very important chapter. It is a chapter which will actually make you test if you really like botany, because it is a chapter which you cannot by heart. It is not a chapter which you can just on the go. It is a chapter which requires you to understand a concept, especially when it comes to cycles, right? Now in today's class, our motivation is very high. Our aim is also very high. Now aim for today's lecture is going to be what? Our aim for today's lecture is going to be introduction to plant kingdom as well as algae. These two things are going to be taught in today's class. So students, if I'm properly audible and visible to all of you, quickly add fire emoji in the chat right now, students. All of you, quickly add fire emoji, students. Fire emoji in the chat. If I'm audible and visible to all of you, students. Hello, hello, hello. Yellow, everyone. Now, all of you, yes, students. My full name is Basavaraj Rajashekar Shirgumpi. My full name is Basavaraj Rajashekar Shirgumpi. That is my full name. Okay, so students, all of you get a bottle of water. Water is very important. We need to drink a lot of water, students, because we don't want kidney stones. Okay. Now, students, Radhe, all of you. Now, let's start the class with one question, students. All of you. Let's start the class with a simple question. Okay. The simple question for today is gonna be, students, is this a plant? All of you, students, is this a plant right now? All of you. Is this a plant? All of you, students, is this a plant? You're like, yes, sir, it is a plant. Is this a plant? All of you. Is this a plant? You're like, yes, sir, it's also a plant, but it is not a plant. You'll be confused. You're like, it doesn't look like a plant. Here, a plant has here. A typical plant here has a leaf, it has a fruit, it has a flower, it has a stem, it has a root. A typical plant here has root, stem, leaf, fruit, flower. But any of these features, do you observe here, students? Do you observe here any of the following features? Any of the following features do you observe here? You're like, no, it is not. I cannot see a root here. I cannot see a leaf. I cannot see a flower nor a fruit. So is this a plant? Yes or no? All of you. Yes or no in the chat right now, students. Yes or no? Now, let's say, is this a plant? Here also I cannot see leaf. Here also I cannot see root. Here also I cannot see a flower or fruit. Nothing. But is this a plant, students? Is this a plant, all of you? Yes or no, students? Is this a plant right now? Okay. We don't know. Let's see. Is this a plant? Okay. It has a leaf-like structure. We have a root-like structure. But I do not see any flower, no fruit here. Okay. This one, students. This one. Okay. Let's see last. This is a gymnosperm. It has a stem. It has a flower. It has a seed. But doesn't have a fruit. Students, all of these pictures which I showed you right now, all of these pictures they might not have roots. All of them might not have stem. All of them might not have a fruit or a seed. But every single picture of a plant till now have one thing in common. What is that? Students, what is that, all of you? What is that one thing which is common? What is that one thing which is common in every single picture I showed you till now? That is the, which color, all of you? Which color? Which color? It was the green color. Every single picture till now did have the green color in them. Everywhere. Every single one of them had a green color. Now, if all of them have green color, it means that they can do a process called as what, students? They can do a process where a plant can prepare its own food. The process called as photosynthesis. So all of these images are plants. All of them can do a process called as photosynthesis because of a green color pigment called as, students, anyone? A green color pigment called as, all of you? Tell me, students, what is that pigment? Called as chlorophyll A. The chlorophyll A is a common pigment, students. Chlorophyll A is the common pigment in all of them. But students, do you think there are other pigments apart from chlorophyll A? Yes, there are. There are something called as chlorophyll B, chlorophyll C, then chlorophyll D. There is carotenoids, there is xanthophyll, there is phycoerythrin. There are other pigments also present. So we will discuss in this class what are the other pigments, what are the other types of your differences among all of them, students. Now, with this being said, shall we start the class, students? Shall we start the class today? This was just the outer hype. Okay, hype is done, students. Hype is done. Now, once the hype is done, students, all of you know the nutrition, all of you know they can do photosynthesis. Now, let me ask all of you one simple check of your knowledge. Okay, tell me, students, kingdom Monera, do you think they have eukaryotic cell or prokaryotic cell? Kingdom Monera has what? Kingdom Monera has prokaryotic cell. Kingdom Protista onwards, every single one of students, every single one of them has what? Every single one of them has eukaryotic cell. So obviously speaking, plants also have eukaryotic cell, students. So if they have, students, if they have eukaryotic cell, do they have nucleus? Well-defined nucleus, well-defined nuclear envelope, Golgi bodies, everything. Yes, they do. So plants also have them. Plants also have them, students. So they are eukaryotic cells. And students, and some of the plants can be unicellular, like your Chlamydomonas and everything, right? But generally speaking, they are multicellular in nature. Generally, students, they are multicellular in nature. Okay, students. So if you ask me what is a plant, so I will tell you, students, plant is an organism. Plant is a organism which is eukaryotic in nature. It is going to be generally multicellular in nature. It is going to be, students, it is going to be what? Multicellular in nature. It is going to have chlorophyll pigment. It can do because of pigment, can do something called as photosynthesis. Photosynthesis. That is your, student, that is your pigment. Okay, that is your pigment, student. That is your pigment. That is your chlorophyll is a pigment. That is a plant, student. That is a plant right now. Now, student, let me ask you. There is a basic difference between your plant cell and animal cell actually. Like for example, if you look at a plant cell, outside the plasma membrane, it has a one more layer. Who can tell me what is that, students? It is found only in the case of plant cell. That is, outside the plasma membrane, there is one more layer, students. What do you call that? All of you. That is called as what, students? That is called as cell wall is present, students. Cell wall is present, all of you. Okay, there is cell wall is present there. Okay, so we have cell wall in the case of your plants. Okay, students. Now here we have your 5 kingdom classification, students. Yes, students, I'm from Bangalore. Okay, now students, here is your 5 kingdom classification right now, given by who? Students, who is the scientist? Students, who is the scientist who gave the 5 kingdom classification? R.H. Whitaker. So R.H. Whitaker gave the 5 kingdom classification: kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, kingdom Animalia. In today's class, we will be focusing on kingdom Plantae. Now, so students, we'll start off with basic history now. We can't start with algae directly. We will start with basic history. Now, for this, I need a volunteer. Raise your hands. Who wants to volunteer? For this, I need a volunteer, students. I need a volunteer for my class. All of you, quickly smash the like button, all of you. All of you quickly smash the like button, and I need a volunteer. Okay, let's take Joshua. Okay, let's take Joshua. Joshua is our regular student. Okay, now students. Now let's say Joshua is walking on the road. Now Joshua is walking on the road right now. Not now, millions of years ago. Okay, let's say during Neanderthal man time, around thousands of years ago, right? Now Joshua is walking on the road, right? Joshua is walking on the road, and he sees around, and he notices some plants around him. Now he will eat a plant. He will collapse. Now that is called as poisonous plant. Now his friend will come. The friend is Suresh now. Suresh is like, bro, you try this. He'll give one more new plant. The new plant will cure Joshua. That is medicinal plant. Now let's say Sharmita wants to build a house. She will cut a tree. So basically, students, I'm telling you, the earlier classification, the old classification, can I tell it was based on? It was based on use and disuse. Whatever is useful, you will give it a name. So medicinal. So we got medicinal plants, that is your edible, generally medicinal plants, poisonous plants, edible plants. So based on the use and disuse, we got this type of classification. Students, we got this classification. Now students, now we have Darshini now. Okay, let's say we have Darshini now. Now Darshini is walking on the road after some time. Now Darshini will notice one plant is this tiny plant. Now one plant is this big. One plant is very tall, taller than her. So what do you call the classification, students? The classification done by Darshini here based on the height of a plant or the size of a plant. Anyone who can tell me? This classification was done by Carl Linnaeus also, students. This classification was done by Carl Linnaeus also. That is basically herb, shrub and tree. That's all, student. That's all. We have the herb, shrub and tree. Now, herb, shrub and tree classification was based on the size of the plant and also the stem of a plant. If you notice here, the herbs have herbaceous stem. Students, herbaceous stem is green or brown? Herbaceous stem is generally green color stem, green color weak stem. Okay. Now, shrub is much more bigger right now. It is much more bigger. Okay. Now, trees obviously very tall, brown color stem, trunk they have. Now, apart from the stem and the size of a plant, we also classified these plants based on the lifespan also. Lifespan. Based on the lifespan also. Or how often they can complete, how much time they can complete their life cycle. Now for example, all of these herbs will finish their life cycle in 1 year. They are called as annual plants. These plants can generally finish their life cycle in 2 years or more than 2. So 2 years is called as, 1 is called as annual, 2 is called as biennial. Now students, if trees can complete their life cycle in many years, that is called as perennial life cycle. Perennial. So many years, 2 to few years and 1 year. So annual, biennial, perennial. Okay. So annual, biennial and perennial, students. Perennial. Now students, now, if I classify plants just by looking from the outside, like you like a person by looking at their face. Oh, nice nose that girl has, a nice eyes that girl has. So if you look at a person, you like them only from looking outside, morphology, external morphology, not true love and all. Okay. Now if you classify plant by looking just from the outside, can we call them artificial classification? All of you, artificial or natural? It is not natural. Natural is truly heart inside, from heart. Naturally is heart. Okay. So classification, students, classification based on external beauty, external morphology, students, external morphology, that is called as artificial system of classification, students. In artificial system of classification, we'll base it only on one or a few specially chosen morphological features. That is from the outside, face only. You're looking at the face, you're not looking at the heart only. Look at the heart, no, sometimes. Poor boys. Okay. Now students, now. Okay, now. That is called as artificial love here. Okay, artificial love. Now, this artificial love was given by none other than Carl Linnaeus actually. Carl Linnaeus, okay. Artificial system of classification. Carl Linnaeus. All of you namaskar quickly. Students, do you remember? Carl Linnaeus was a scientist who gave us what? Binomial nomenclature, taxonomical hierarchy. All of it was given by the scientist only. Please remember, pay the respects. Respect. Put the name, some respect here. Okay, Carl Linnaeus, students. Mangifera indica, everything, binomial nomenclature, taxonomical hierarchy, everything was given by Carl Linnaeus, students. Everything was given by Carl Linnaeus. Okay, Carl Linnaeus, students. His main classification, was based on the stamen. Students, you might have heard, no. If you have done a chapter called as morphology chapter, you might have heard about something called as syngenesis condition, synandrous condition, monoadalphous condition, diadalphous, polyadalphous. All that was done by Carl Linnaeus only. All that was done by Carl Linnaeus, students. Carl Linnaeus only. Carl Linnaeus only. Starting. Okay. Now, students, now we are like, no, we will not do something called as artificial classification. We will not do something called as artificial system classification. We will look at the inner beauty now. Now, students, now there are two scientists. Now there are two scientists who looked at the inside heart, no, inside the plant. Okay. So we have George Bentham and Joseph Dalton Hooker. So Bentham-Hooker classification. Bentham-Hooker classification, students. Bentham-Hooker classification is based on the internal structure. Students, it is based on the ultrastructure, that is the inside, how it looks. Inside, students, inside, fine structure of the cell. Does the cell have eukaryotic, prokaryotic cell? Does the cell have cell wall? What organelles are present in a cell? Classification based on that. Let's say we have a plant which does not have chlorophyll, parasitic in nature, Cascuta. So the classification started now. Okay, the ultrastructure, anatomy. Let's say one plant has anatomy is changing. Monocot has different anatomy, dicot has different anatomy. Anatomy of gymnosperms is different, anatomy of angiosperm is different, anatomy of bryophyte is different, anatomy of algae is different. So anatomy came into picture. Then we had embryology also, development of embryo stage. In monocot we have one cotyledon, dicot we have two cotyledons. So embryo came into picture. So embryology, phytochemistry, that is chemicals obtained from plants. So when we look at, students, when you look at ultrastructure, when you look at anatomy, when you look at embryology, when you look at phytochemistry, when you look at the internal structures, internal beauty, students, what do you call that? Students, what do you call that, all of you? What do you call that? Internal beauty, all of you. Natural or artificial again? We call that natural system of classification. Natural, true love, this is true love here. Natural system of classification. Okay, natural system of classification, students. That is classification based not just external but also on the internal features. Also on the internal features, students. Now, students, I will tell you one drawback. I'll tell you one drawback. All of you will relate to it. Let's say you like a person for their face. As the person grows older, will the face change? Yes, obviously everyone grows older, face changes. So you'll stop loving them, huh? You can't do that. So that's why it's called as artificial. That is what's called artificial. So even in the case of plants, if you try to classify something based on morphology, will the morphology change? Let's say one plant is identified by the leaf. In winter, all leaves are gone. Students, in the winter time, all the leaves are gone. So how will you identify them, students? How will you identify if all leaves are gone? Let's say in non-flowering season, there is no flower only. Can you identify them? No. So that is the main drawback of artificial system of classification, because the features are there. No, in artificial, they can change. They can change. That is the main drawback, because anatomy will not change easily unless there is evolution. Okay. That's what it is now. So there is a comparison here which I will give you. Students, here is a quick comparison. Here is a quick comparison here. Artificial, natural, Linnaeus, Bentham and Hooker. Here only morphological features. Here we look at the internal features. Ultrastructure, anatomy, embryology, phytochemistry, students. Any doubt here? Students, any doubt here? All of you. Any doubt here between these two? Now, students, now, how can we talk about biology and not include our Charles Darwin? Yes or no, students? Yes or no? How can we talk about biology and not include Charles Darwin? Right. So Charles Darwin is like, all this is okay for me, right? All this is okay for me, but you also include evolution also. You should also include evolution also. So with the students, with that, okay, with that, we got some students. We got the phylogenetic system of classification. From Charles Darwin, we got the phylogenetic system of classification. We got the phylogenetic system of classification. Now, in this phylogenetic system of classification, there is also use of evolution. Students, अगर मैं हिंदी में बात करूंगा तो आप पागल हो जाओगे. ठीक है, मेरी हिंदी उतनी अच्छी नहीं है, right? Now, students, that is the main thing about Hindi. I can't talk Hindi very well. I'm trying still. Okay. Now, this phylogenetic system of classification is based on evolutionary relationship. Okay. Now, what do I mean by that, students? What do I mean by that, all of you? Oh, already giving your respects, huh? Okay. Now, students, now, students, let me get the thought back. Okay. Now, students, have you noticed there are different types of mangoes? All of you? Have you noticed there are different types of mangoes? Different types of mangoes? All of you? Have you noticed or not? There is Totapuri mango, Alfonso mango. There is different types of mangoes. Yes or no? There are different types of oranges also. Red orange, big orange, small orange. See, I don't know many of them. That's why I'm telling big and small. There are varieties of mangoes also. Now, students, if you try to make a connection between them, can you make a family tree of them, students? Can you make a family tree that this mango was there, from this mango came, this mango's daughter is this mango, this mango's mango, one more mango came. Yes or no? Now that is called as family tree of mangoes. Now there you use phylogenetic relationship. Okay, so here are the different type of oranges. Okay, so this type of classification is called as phylogenetic system of classification. It is based on evolutionary relationship between the organisms. Phylogenetic. Lemon. Students, have I told you behind the story behind lemon? Students, have I told you behind story behind lemon? Why I call it lemon and not lemon? Students, have I told you? Why do I call it lemon? This is a nice story, students. Listen to me. There was a friend of mine who teaches in a very big institute right now. Big institute, right? When we were classmates, he was from Kerala, okay? And when we were in MSc, right? When we were in MSc first year, I got lemon rice that day. Lemon rice to the MSc first year. I got lemon rice tiffin box, and for some reason he had never told. No one has ever corrected him also, right? He's like, bro, lemon rice, lemon rice, and we're like, what? He's like, lemon, and from that day onwards, this was the second week of MSc, second week of college. From that day onwards till the graduation, we have always teased him by lemon. And after that, we all of us stopped saying lemon only. We never used lemon. Like now in my class, I used lemon. But generally we say lemon, right? Lemon forever because of him. And students, you should realize, because of one time he said lemon by mistake in front of us, it became a tagline for him. It became a tagline. We used to tease him all the time. A lemon boy. Okay. So here we have lemon, we have mandarin, we have sweet orange, we have blood orange. All because of phylogenetic, students. All because of phylogenetic. Okay. Now, students, how do we find evolution? Like, how do we find the evolution traces, right? How do we do it? Right. Students, don't call me bully now. Bully. Okay, it's lemon. Now, students, right? How do we find the evolution? Let's say we have Dryopithecus, Ramapithecus, Australopithecus, Homo habilis, Homo erectus, Neanderthal man, and Homo sapiens. Like, how did we even manage to get a chronology? How? Cash is a little sick. Students, how did we get that chronology? By fossils? Yes or no? So this can also be done by fossils. Here also we have fossils here. So this mystery can be solved by fossil records. Now, but what if there is no fossil? What if there is no fossil? In that condition, what do we do? Students, if there are no fossils at all, how do we find the connection? How do we get the missing links? The missing links, if there are no fossils, can be found by other methods. Now, what are the other methods? Other methods are this is plant fossils. The other methods are your numerical taxonomy, students. One of the examples is your One of the example is what? One of the example is numerical taxonomy. Numerical taxonomy. So with the help of numerical taxonomy, we can find the relationship, okay, of different organisms. Now this is how numerical taxonomy looks like. Numerical taxonomy looks like this. Now in numerical taxonomy, we take different types of your plants. We take 10 different characters. Now let's say this plant has this character. We use 1. So 1 means present, 0 means, students, binary, right? 1 means present, 0 means absent. So if this character is present in this plant, 1, 1, 1, 1, 1, 1, it is present. This character is absent, so 0. Similarly, we do that 1, 0 method for every plant and every character. We get a data. And again, students, here all of these data is equally treated here. Now we take this data, students, we feed into a machine. We take a machine computer, give the data there. Now once you give the data, the machine will give you a nice list here. Now for example, I'll show you a list here. Students, see here. Now let's say this is the data which we get, initial data. Here we can see, students, here we can see all of you. In A and B plant, in plant A and plant B, right? Plant A and plant B, we can see 1, 1, 1, 1, 1, 1, 1, 0, 1, 0, 1, 1, 1, 1, 1, 1, 1, 0, 0, 0. So can you see by this data, can you see plant A and plant B, they have some similarities among them? They have some kind of evolutionary relationship among them? Yes, they do. Yes, they do, students. Now similarly, if you see C and D, 1, 1, 1, 1, 1, 0, 1, 0, 0, 0, 1, 1, 1, 0, 1, 0, 0, 0, 1, 0. There are some similarities, differences are also there. So with the help of that, okay. Now students, look at DNA. Difference. Look at DNA now. 1, 1, 1, 1, 0, 0, 0, 1, 0, 0, 1. All zeros here. So do all of you notice D and E share some similarities. A and B share more similarities. So we can find a connection among them. Connection between them. Now, similarly, A is somewhere connected somewhere to B and C. So with the help of this data, right, we can find something, we can make charts like this. They're called dendrograms and cladograms. We can make many of them, right? So based on similarities and differences, which we give the data to the computer, after making something like this, we give this data to the computer will give us this. Computer will give us this. And with the help of this, we can find the relationship between different organisms. That is your, in the use of numerical taxonomy. Use of numerical taxonomy where? In phylogenetic system of classification. In the phylogenetic system of classification. You understand, let's say here we have two plants, plant A, plant B, plant C, plant D, plant T. Now what do? Plants, characters. If this plant has this character, we write 1, 1, 1, 1, 1, 1, 1. If this character is absent, we write 0. We take this data, whatever we type, right, characters and the plant, we take this data, we give it to computer, and computer will generate this. Now based on this information, I can tell, this plant and this plant, they have some connection. This plant and this plant, they have a common ancestry. Yes or no? Now from this plant, we got new plant later on. Now with the help of this, we can do the phylogenetic system of classification. That is the whole point, right? The whole point of phylogenetic is to find the connections. Okay, that's what I'm saying. Apart from numerical taxonomy, there is something called as cytotaxonomy. In cytotaxonomy, we use chromosomes number, chromosome structure, chromosome behavior also. Why? Because students, why? Because chromosomes will not change eventually. They'll remain the same generally unless there is a mutation. They'll remain the same. So we can use cytotaxonomy. We can also use the chemotaxonomy also. The primary metabolites, secondary metabolites released by the plants. So whenever there is fossil is absent, we can use all of these things to find the phylogenetic system of classification. To make the phylogenetic system of classification, we can use all of these whenever there is no fossil record. Clear, all of you? Clear or not? All of you clear? Can I get a thumbs up in the chat? Students, can I get a thumbs up in the chat? Thumbs up in the chat, all of you. Can I get a thumbs up in the chat, all of you? This is your entire phylogenetic system of classification, phylogenetic system of classification. Okay, so we learned about artificial, natural, phylogenetic. 3 different types of classifications today. No, students, no. Now here also we have the current system of classification given by your Eichler. Here we have cryptograms, phenylograms. Cryptograms we have thallophyte, bryophyte, pteridophyte. 3 different types of phyte here. Yes, thallophyte, bryophyte, pteridophyte. Phenylograms, gymnosperms and angiosperms. Phenylograms have seeds. Cryptograms do not have seeds. Simple as that. There are no seeds here. Here we have seeds. Okay, seed and no seed. Okay. Now students, here is your final verdict on plants. Eukaryotic, cell wall, multicellular, photosynthetic, plantae. Now students, now with all of this done, shall we start algae now? Students, shall we start algae? I know all of your DNA students are here. You are undergoing revision process now. All of you are undergoing revision process. Yes. All of you. With this being done, shall we start with the algae right now? First part is done. Students. All of you. Shall we start? All of you have become greedy now. 2 times you're listening to my class. Huh? Students, shall we start algae? Yes or no? Shall we start? Okay. Okay, students, okay. Students, how is your confidence now? How is your confidence in the early part? Ultimate confidence. Now, I'll finish algae in one flow. Right. After that, I'll show you some NCRT lines. I'll show you. You'll feel better. Okay. Students, now. Let's start with algae, students. Let's start with algae right now. Okay. Let's start with algae. Okay. Now, when I talk about algae, students, here is a comparison of algae. Here is a, students, here is a comparison of algae right now. Now, students, do you think they have similarities? Here I have root. Here I do not have no root. Here, no root. Here we have no stem. Here we have no leaf. Here in the case of your plant here, we have no root, no stem and no leaf. So this structure is actually called as thalloid structure. A thalloid structure is a structure where there is no root, no stem, no leaf. That is thalloid structure. So this is what an algae looks like. This is what an algae looks like, students. Okay, that is called as thalloid structure. That is called as a thalloid structure. That is called as a thalloid structure. There is no root, no shoot, no leaf, no flower, no fruit, no seed. Still it is a plant, students. Still it is a plant. Still it is a plant. That is called as a thallus structure or thalloid structure. Thallus. Thallus or thalloid structure, students. Okay. Thallus or thalloid structure. Undifferentiated body. Very good, Sharmita. Undifferentiated body here. There is no root, no shoot, no leaf. Thallus. Now, students, since they were called thallus, they were placed under thallophyta. So all the algae were placed under thallophyta. Algae under thallophyta, students. Now, would you think there were other plants also initially in thallophyta? Other organisms in thallophyta? Yes, students, yes, they were. All of your fungi, thallophyta. Algae, thallophyta. Lichens, also thallophyta. So all of these organisms were under thallophyta in the starting. But as we understood the different plants and organisms, we removed fungus from here. We removed the lichens from here. Fungus got its own kingdom. Lichens was the ignored child. Lichens we ignored only. Okay. So right now, students, right now, the only member of your thallophyta, the only member of thallophyta is who, students? Who is the alone member? Students, tell me. Who is the lone warrior here? Who is the alone, lonely warrior of your thallophyta, students? Who is it? That is your algae. Algae is the only member right now. Algae is the only member of thallophyta. Now, students, now, if you want to give a definition for students, if you want to give a definition for algae, algae definition is very simple, students. Algae are chlorophyll bearing, they have chlorophyll, simple, thalloid. Thalloid means they do not have anything like a root, stem and leaf. Autotrophic in nature, that is photosynthetic, they can produce their own food. They are largely aquatic, both freshwater as well as marine organisms. That is the definition of algae. That is the basic definition of algae right now. Simple, thalloid, chlorophyll bearing, photosynthetic in nature, organisms. Organisms. Now, question, students, how a question arises? Now, question, students, question arises. How do you think algae are classified? Anyone who can tell me the answer? Students, how do you think algae are classified? Anyone? Anyone, students? Science world? Yes? Angela, Supriya, Sharmita, Joshua, tell me. Students, how do you think algae are classified? Do you think they're classified on, can we classify them based on shape? You cannot, because there are variety of shapes. There is your unicellular, filamentous, colonial, membrane-like, like paper-like structure. There are variety of shapes. You cannot classify them. Can we classify them based on their habitat? No, because habitat, there are a variety of habitats. They can be present on rock, present on your soil, present on wood, present on your sloth bear, present on lichens. So they're present almost everywhere. So we cannot classify them based on their habitat also. Very difficult to classify. Can we classify based on nutrition? No. Main nutrition is autotrophic in nature. So nutrition is also no. Can we classify based on reproduction, students? Can we classify based on reproduction? All of you? No. Because they have vegetative, like binary fission and fragmentation. Vegetative is present here. Asexual is present here. Asexual by spore formation. Sexual actually is 3 types here. Students, sexual actually is of 3 types here. 3 types. Students, remember, in asexual we can also find zoospore also. Asexual we can find zoospore also. Zoospore. Sexual is of 3 types here. That is isogamy, anisogamy and oogamy. Now, what is the difference? Understand. Very simple difference. All of you tell me, this motetale and this motetale, do they look like a same? Do they look like each other? Students, do they look similar here? Iso means what? Iso means same. Iso means same. Gamy means gametes here. So in isogamy, 2 gametes look alike. Same size here. Same size. Both are same size. Now, what about anisogamy, students? In the case of anisogamy, the 2 gametes are different from each other. Anisogamy. Now, what about, students, oogamy? Oogamy. The main difference here is that, yes, the sizes of your 2 gametes is different here. But apart from that, students, here, the female gamete is large here. And the male gamete is very small. Just like humans. In humans also, the egg is very large. And the sperm is very tiny. Now, if you notice here, students, the small gamete, the male here, is generally motile. It has flagella here. While the female is large and non-motile. Small and motile in nature. Small and motile, that is the male. Small and motile, female is large and non-motile. Very simple difference between the all of them. Iso and iso, students. Now in examination, what they'll students ask you? In examination, they'll ask you the examples, mainly examples. Okay, students, what are the examples here? Please understand here the main famous example which has always been asked is your isogamy, which is motile, which is non-motile here. Now under isogamy, there are two examples mainly. Two examples. Okay. That is, gametes can be motile. Example is eulothrix, motile, and non-motile example is Spirogyra. So here, please remember, there are two examples to remember. Example for motile and example for non-motile. Motile and non-motile, students. Motile and non-motile. Motile example is eulothrix and non-motile is Spirogyra. So if you remember this, you are sorted. You're sorted, students. You're sorted here. Okay, Spirogyra. Now, anisogamy, Eudorina is an example. Oogamy, Fucus as well as Volvox. Fucus and Volvox are two different examples of oogamy. So, iso, aniso, oogamy, this you have to remember at any cost. You have to remember all of this, students. You have to remember at any cost. Okay. Now, students, if you cannot classify algae based on the shape, you cannot classify based on habitat, cannot classify based on nutrition, cannot classify based on reproduction. So how can we classify, students? How can we classify? Students, tell me, students, tell me. How can you classify? Tell me, students. How can you classify? Anyone? Who can tell the answer? Students, who can tell the answer? How do we classify them? What is the main best way to classify all the algae? Pigment. That is going to be the pigment, students. Pigment. Because there are three main pigments. There are some are not given your NCRT, right? The main three exhibit major pigments are your red, green, and brown. Red, green, and brown are the three different types of pigments here. Three different types of pigments, students. Pigments here. So based on the pigment, there are three different names here. One is called as your chlorophyce is green color, pheophyce is brown color, and rhodophyce is red color. So based on the pigment, there is a classification done here, students. Classification done. Pigment, students. So repeat after, students. Repeat after me. Chlorophyce, pheophyce, and rhodophyce. Three different types of pigments present here. Now based on this pigment, there are multiple examples under each one of the students. Under each one of them, different examples are there. Okay. Now before we go ahead, students, now let's start to discuss a few general features or the characters of each type of your class of algae. What type of cell wall do we find in green algae? What type of cell wall we find in brown algae? What we find in red algae? Let's start, students. Let's start, all of you. Okay. Let's start, students. Let's start. Okay. Now, first things first, students. First things first. In the case of your algae, do you have plant cell or animal cell? We generally have plant cell. We generally have the plant cell. So if we have plant cell here, let's say this is a cell right now. This is a plant cell right now. This plant cell, let's say this is the plasma membrane. Around the plasma membrane, students, we have something else. What do you call that, students? Around the plasma membrane, we have something called as the cell wall. Around the plasma membrane, we have the cell wall here. Now, students, in the case of certain algae, right, certain algae, even outside the cell wall, in the outside of the cell wall, there is one more layer here. it is like a moisturizer. Around the certain algae, there is a moisturizer. Can anyone tell me, students, what is this moisturizer, or anyone? Yes? Anyone? All of you. What is this moisturizer around the cell wall in the presence of algae? Tell me, Darshini, tell me the answer. Darshini, tell me the answer. Tell me, what is this? Anyone? What is this, students? What is this? This moisturizer is called as hydrocolloidal substance, students. Hydrocolloidal substance here. Now, this hydrocolloidal substance is what? It is basically a chemical deposition. It is a chemical deposit here. Chemical deposit. Now, students, understand what is this deposit. Understand, very simple rule, very simply explain, students. Okay. Let's say there is a algae. Algae are mainly found in the case of your aquatic ecosystem. Now, in aquatic ecosystem, especially in the case of oceans and seas, right, there is something called as high tide and low tide. Yes or no, students? There is something called as your high tide and low tide. That is based on the moon's gravity. Based on moon's gravity, there is high tide and low tide. Now, whenever there is low tide, this algae gets exposed to sunlight. Whenever there is low tide, the algae surface gets exposed to sunlight. Now, students, you tell me, if the algae is there, right? Algae surface, if it gets exposed to sunlight, can it undergo dryness? All of you, it can undergo dryness. Now, algae is very smart. Now, algae will make sure it doesn't go dry. What algae do is that algae has something called as hydrocolloidal substance. This hydrocolloidal substance will absorb the liquid, absorb the water. Okay, it will basically hold water. It will hold water. If it is able, students, if it is able to hold water, it is like a moisturizer on the algae now, because it is completely water is there present on the top. So if water is present on the top of it, it will not go dry. Yes or no? It will not go dry. That's all. So that is the main function of hydrocolloidal substance, students. Okay, hydrocolloidal substance. Okay, now students, now apart from this, all of you need to understand what is the cell wall made up of here, because cell wall deposition is different. Okay, now let's talk about one by one what happens in the case of your green algae, brown algae, and red algae. Students, if you know the answer, start spamming the answer. Now, in the case of your green algae, what do we have, students? In the case of green algae, what do we have? In the case of normal green algae, brown algae, and red algae, one thing is which is very common. It is a type of your polysaccharide which does not have helicals. Does not have helicals. The one thing which is common is called as cellulose. It is going to be common. No. Students, apart from cellulose, in the case of green algae, it also has something called as pectin. In the case of brown algae, it has a hydrocolloid called as algin. In the case of red algae, it has something called as carrageen. Carrageen, students, carrageen. Okay. Carrageen plus pectin plus polysulfated esters. Polysulfated esters in the case of your red algae. Now students, this is what you should know, right? Karachi, right? That's fine, students. Karachi will be there. Okay, clear all of you students? Clear? Clear all of you? Yes or no? Okay. So that is the basically your cell wall in the case of three different algae here. So cell wall, hydrochloride substance, plasma membrane is present on the inside, water holding capacity by your different types of structures. Now students, what about flagella? How is the flagella different here, students? Now flagella is different in the case of your brown algae, green algae, but red algae, I'll tell you later, students. Okay. Now students, what is the main function of flagella? The main function of flagella, can I tell it helps mainly in motility? Yes, it does help in motility. It does help in motility. So again, we'll write green algae first. Okay, students. In the case of green algae, students, please understand, this outer part is called as what? Apical part. From this apical part, two flagella will come like this. You might have seen in Chlamydomonas. From this apical part, they'll start to come like this. They'll come like this, students. Okay. So they'll come like this. That is present in the case of your green algae, students. Green algae has something like this. From this apex, two flagella will come out like this. Can you see? Now, here we have equal flagella. Both flagella are equal here, students. Equal flagella. Can you see? They're equal right now. Both of them are equal right now. Equal. Okay. Equally arranged, and they are also in the apical. Equal and apical. Sometimes the number can increase between 2 to 8 also. 2 to 8 in number. Okay, 2 to 8 number. Now flagella can vary. Now students, in the case of your brown algae, here the gametes have something called as pyriform shape or pear shaped. They look something like this. They look something like this, or they look something like this. The gametes here. Now here, how is the flagella, students? Flagella here, it is arising laterally, and if you notice, students, these flagella are almost unequal. All of you? Yes or no, students? Yes? All of you? Understanding? Apical means top part. Top part is apex from here. When flagella will come. That is called apical. Apical is this top part. Apical means top. Apical meristem, top part. From this apex flagella comes. This top part is called apical part. These are lateral, students. Again, they are two in number. They are unequal, and they arise from lateral here. Now this is called as pear shape, also called as pyriform. So that is your basically your flagella. Now students, what about red algae? Students, please understand. In the case of red algae, flagella is absent. So please, students, please remember this. Okay, done all of you? Done? In the case of green algae, equal apical 2 to 8. Brown algae, pear shaped, your gametes are two unequal lateral. Red algae, it is flagella is absent here. Completely gone right now. Okay. Students, sound check. All of you, sound check, sound check. Sound check. Sound check. Perfect. Okay. Perfect. Perfect. Now, students, here we can see in the case of green algae, red algae, and brown algae, right? In all of these algae, right, students, all of these algae, right, one pigment is always common. So, which is that pigment? The common pigment in all of them. The common pigment is going to be what? The chlorophyll A. Chlorophyll A is going to be common in all of them. Now, students, apart from chlorophyll A, here is a small trick for all of you. Small trick. B, C, and D. That is a common, student. That is a common pigment in all of them. B, C, D. Now, students, here green algae enough, but red algae, students, red algae, right? Red algae has a pigment for the redness. For the redness, students, for the redness. Can anyone tell me what is the red pigment called as? It starts from phi. Students, anyone? Yes or no? All of you. Something called as what? Students, phyco, something with red. What is red called as? Phycoerythrin. So in the case of red algae, we have something called as phycoerythrin. Now students, here in brown algae, we have something called as fucoxanthin actually. We have something called as fucoxanthin. Now students, please understand here, this fucoxanthin is there, right? It can, if the fucoxanthin is very high, or it can be very low. When the fucoxanthin is very high, it will look very brown color. When the fucoxanthin is very low, it will look green or olive green. It will look green or olive green. So students, depending upon the concentration of fucoxanthin, and there is obviously carotenoids, there are different carotenoids also, carotenoids are there, right? This entire look of algae will change. It can be brown completely or green completely, depending upon the concentration of the pigment there. Okay, that is a brown algae. Now students, all of these algae, they prepare food. Yes or no? They need to store the food somewhere. Okay, they need to store food somewhere. Okay. So what is the food actually? What is the food actually? Like they need to store, right? What is the food stored as? Now again, same thing we do, students. We do the same thing here. We do the green algae, brown algae, and red algae. Okay. Now students, now in the case of green algae, it is very, very simple. It is stored as starch, nothing complicated. Starch can be there. There can be oil droplets. Oil droplets can be there. But in the case of brown, it becomes complicated. There can be some complicated carbohydrates also. Something called as mannitol and laminarin. Okay. Oh, I think I missed out, students. I think I missed here. Oh, one second, students. I think I wrote ulta here. One second. ABC, no, but it has to. I written red algae up. 1 second. I'll just change this something. 1 second, students. 1 second. Usually I always write brown, green, brown, red. No, I wrote this one, right? So this will go now. This will become C and this will become B now. Unfortunately, this will become your A, B, C, and D. Okay, students, just make the mistake. Just change this. So just change this because usually I write A, green, brown, red everywhere. Here I wrote red on the mistake. I wrote everywhere. Just check once again. Green, brown, red. Green, brown, red. Okay. This was jumbled here, so I was teaching you that. Okay. Don't worry, students. Please don't worry. Honest mistake can happen. Yes, honest, because I was in the flow of like how I always teach. Right. Okay. Thumbs up, all of you. Thumbs up in the chat. Thumbs up in the chat, all of you. Clear. Okay, students. Now they're going in the flow now. Yeah, brown students here, brown algae. In brown algae, we have the mannitol and laminarin. In the case of red algae, we have something called as from Floridia, Floridian starch. Now, the best part of this students, Floridian starch is that the structure of this Floridian starch, right? The structure of this Floridian starch, okay? The structure of this Floridian starch is very similar to something called as amylopectin and glycogen. Very similar to mylopectin and glycogen. Okay, glycogen right now. Okay, Floridian starch. Students, still here ain't out. Because if after this algae is over, 15 minutes of class is done. 15 to 20 minutes, 20 minutes max to max 20 minutes class is done today. Algae is done. Students, any doubt till now? Students, do you want these notes? Students, do you want these notes? Students, DNA students will have all the notes, no? All the DNA students have the notes, yes or no? All the DNA students have the notes. They have the notes already. I think by mistake I've given some other notes for DNA students. By mistake I gave some other notes. So please check that once. I think I've given you wrong notes in DNA batch for this one. Right. So, right. So I'll give you these notes also. I will combine all the notes and I'll give you. Okay, students. Students, I will ask you questions orally. A lot of questions I'll ask you orally. Okay. What is RT? RT2. What is RT2? Yes, sir. Okay. Students, I've given the notes in Telegram. You have seen, right? All the notes have, don't spam. Okay, students. Now, we have the pigments done. All the cell wall done. Green, brown algae, mectin, algin, carrageen. Flagella done. Pigment done. Storage done. Students, this is all is not required. Okay. Students, I give you, look at the different shapes here, all of you. All of you. Look at different regular test. Oh, regular test. 550 is a good score. 550 is a good score. 550 is a good score, don't worry. So here, unicellular algae, Chlamydomonas. Colonial is Volvox here. Filamentous, Pyrogara, Ulothrix, filamentous. Students, which one is motile and which one is non-motile right now? All of you. Which one is motile and which one is non-motile right now? Students, smash the like button, tell me the answer. Smash the like button, tell me the answer. Which one is the motile, which one is non-motile here? Motile, non-motile. Quickly, students. Which one is motile, which one is non-motile? All of you. Yes. Tell me which one is motile, which is non-motile. All of you, quickly. Motile, non-motile, one. Now, students. Now, multicellular, example is Cara, one example is that. Students, in the case of your green algae, right, there is something called as pyrenoids. Now, these pyrenoids will help in storage. They also help in storage. Protein can be stored there. Okay. Now, students, if you look at chlorophyce, there are different types of your chloroplast. So, chloroplast will vary a lot. Students, I always tell this. This was one of my 10 marks question or 5 marks question. I don't remember. In my examination, experiment examination, practical examination of algae, students, by the way, I had a 100 marks paper for algae. Students, if you notice, my tongue is twisting because this is my fourth class for today. Right. By the end of day, my tongue is twisted now. Okay, continuous talking now. Students, this was I had a 100 marks paper for algae. Just algae, 100 marks. We had around 40 different algae to remember. 40 different algae, 100 marks paper. This was one of the practical exams. Right, where they would give us, give us a bunch of green algae. Like they'll give one bowl. They'll go. I don't know. My lab attender will go to this one nice. I want to say Mori. I want to say one drainage. But I don't know where he got from. Lab attendant used to go to this drainage. From there, he used to bring one nice lump of green algae, put it on the table, and he's like, go take. He used to tell, go take all the algae. We used to take one forceps, go take one algae from there. One algae we used to take very dirty smelling algae, Take it, keep it on a china dish, separate out the algae and identify them, students. Identify them, right? We used to identify all the different students. We used to identify different algae, green algae, based on the chloroplast shape. Let's say if it is cup shaped, Chlamydomonas, it can be spiral shaped, star shaped, reticulate, ribbed shape. So all of this was done, students. All of this was done by the different types of chloroplast structure. Okay. Someone is asking very serious question. Some serious questions, students, you're asking me. Some serious questions. Now, students, here is a list of all the information you need for green algae. I'll give you exactly 30 seconds to receive this. Then I'll ask you questions. You'll get 30 seconds to look at this image. Habitat, cell. See, students, cellulose is always on the inside. Pectose is on the outside. Please, students, please remember this. Okay. By God promise, I can't say that's BT better or something, butter or something. God promise, I can't tell that. Some BT better. What are you telling me? Now, students, now. Pigments, all of you know, chlorophyll A, chlorophyll B, you know. Starch is pyrenoids, protein plus starch, pyrenoids. Students, here, examples very important. Chlorella, Chlamydomonas, Volvox, Spirogyra, Cara. So please remember these examples. Please remember these examples. You'll do good in life. Okay. Reproduction, all types. Reproduction is all types reproduction, students, here. Okay. Done? My tongue is also hurting. My jaw is only hurting now. Students, if you want jawline in your life, talk for 1.5 hour, 3, 4, 5. Talk 5 hours continuously every day. Students, right? Talk 5 hours completely, you'll get jawline. Right? Don't ask me where is mine now. Okay? Missing. Now, students, this is all the information for chlorophyce. Now, students, now we come to pheophyce. Ready, all of you? Can we start pheophyce now, students? Shall we start pheophyce? Yes. Shall we start? All of you? Yes. Should we start pheophyce, students? Should we start pheophyce right now? Yes or no? Should we start? Or all of you are sleeping already? All of you are sleeping, no? Students, smash it like button, we'll start pheophyce. Students, it will not take much time. Everything is done almost. Everything is done. Okay. Students, pheophyce is called as what? Pink algae. Tell me, students. Pheophyce is called as what? Brown algae, all of you. Pheophyce is called as brown algae. They're mostly marine in nature. See, these are the examples. Ectocarpus, students. Ectocarpus here. Dictyota here. Sargassum. This is called as sargassum. These are different images. These are different images here. See, Dictyota. It looks like one underwater plant. It is technically a seaweed only. It is technically seaweed, students. Technically seaweed only. Okay. Now, sargassum here. Fucus. Fucus is like your, students, fucus is like your paper, like cara for like your alva only, right? It looks like a paper here, paper, which is floating everywhere. Okay. Students, here is all the information which you need for your pheophyce, students. All the information you need for pheophyce. Okay. Students, see here. Found in marine mostly. Cellulose and algin. Remember, algin was your hydrocolloidal. Xanthophyll, that is your fucosanthin. Mantle laminarin. Ectocarpus laminaria, fucus, sargassum, and dictyota. Reproduction, all the three again. All reproduction is found here. Okay, reproduction is found here, students. Students, if you want these notes, you have to join the Telegram. Without the NEET English Telegram, you can get. Now, students, red algae, students. Red algae are called as your ancient algae. They are oldest people in the town. Okay, oldest people. Red algae are the oldest. Green algae is the youngest child. Okay, so students, red algae. Students, red algae is used to eat food also. If you have eaten, students, if you have eaten sushi in your life, if you are rich in your life, try eating sushi in life. Okay, if you're rich, very expensive food, students. One roll, students, nothing it is. One paper and rice they put inside. Okay, now this one will cost you around 50 rupees. 50 rupees. Entire dish 300, 400 rupees it is. 400 rupees. Okay, that is basically nori sheets. This outer part is called as nori sheets, students. Nori sheets are basically, yes, senior citizen. Yes, senior citizen. This nori sheets are basically seaweed. They're basically the seaweed here. Seaweed, students. Now, students, most ancient algae here. Senior citizen, all of you, put some respect to them. Respect here. Ancestors here. Students, Here are the different examples. Beautiful porphyra, beautiful porphyra. So beautiful, so elegant, porphyra. Here we have gracilaria. Looks a little weird, okay? Looks like one straw paper, a red color straw. Then we have gracilaria can be used to make agar agar. Gelidium. This is a big straw here, big straw. Gelidium. Gelidium. Gracilaria gelidium. Okay, they can be used to make agar agar. Here's all the information, all the information. Here you tried cellulose, carrageen, polysulfate esters, everything here. Few phycoerythrin here, Floridian starch here. Examples: Polysiphonia, the beautiful one. Gracilaria gelidium. For students, porphyra is like paper again. Students, porphyra is like paper again. Paper. This is red paper. This is red tissue paper again. Porphyra is like your red tissue paper. Okay, red tissue paper. Okay, so fragmentation, asexual by non-flagellated spore, but here in the case of your rhodophyceae, we can only see oogamy here, only oogamy here, students, only oogamy here, and here non-motile, students. Why non-motile? Students, why do we have non-motile here? Shinchan is very rich. Shinchan is rich people. Okay, we are poor people here. Okay, students, why non-motile? Non-motile because flagella is sleeping. There's no flagella here. There's no flagella. There is no flagella here. Students, with that, this table is easy to understand now. This entire table is done. Yes or no, students? Yes or no? Yes or no? Friends, obviously I know students Shinchan. I grew up watching Shinchan and Himawari. All of you. Table is clear to all of you. Brackish means salt water, somewhat salt and mix of water. Green algae, brown algae, red algae, chlorophyll, storage, cell wall, flagella. Absent here completely. Okay. Yes or no? Yes or no, students? No. Students, I know everyone. I watch everything which you have watched. I have watched almost everything which you have watched. I watch Chota Beam also, Ben 10 also, anime also, football also. I watch everything what you watch. Okay. So nothing is there. Now, students, now I'll teach you something called as your life cycle. I'll teach you something called as life cycle. Right? After this life cycle, I'll teach you something else. Okay? Okay, students? Let's do life cycle. Yes or no, students? Let's do life cycle. Okay, students. Let's say. Now, before I start the life cycle, I want all of you to understand. Generally, right? Generally, plants have two different stages. Every single plant has two different stages. One is called as gametophytic stage. One is called as sporophytic stage. Gametophytic, sporophytic stage. Okay. Now, students, what is the gametophytic stage? Gametophytic stage is always the haploid stage. It is always the haploid stage. While the sporophytic stage is always diploid. Okay. Right, students, sporophytic stage is always diploid. Sporophytic stage is always diploid right now. Okay. Students, state people can obviously watch this because it's already in your syllabus. Okay. It is always diploid. Now, students, remember, tell me, use one brain cell. Use one brain cell you use. Okay, tell me, gametophyte will produce gamete or spore? All of you, use your one brain cell and tell me, gametophyte will produce gamete or spore? Gamete. Sporophyte will produce, students? Sporophyte will produce spore. So, if sporophyte can produce spore, gamete will produce gamete. So, please, students, please remember that. Yes, good morning, all of you. Okay, students. Now, let's do the entire phase of life cycle. Shall we start? Okay. Now, here we have the main plant body, students. Main plant body. This is your main plant body, green in color. And students, remember, in the case of bryophytes and in the case of algae, the main plant body is gametophyte. In the case of your algae and bryophytes, the main plant body is always gametophyte. Students, gametophyte is what? Yes. This gametophyte is haploid diploid. This gametophyte is always haploid, students, haploid in nature. Now, this gametophyte, students, will give you what? Gametospores. It will give you by mitos. No, I'll not tell you. Okay, students, tell me. Here we have gametes here. We have gametes here. Students, these gametes are haploid. This is haploid. So, should I write meiosis here or mitosis here? Students, haploid, mitosis here, meiosis. We have the mitosis here. Okay, fine, I'll tell you. Okay, students. Now, these gametes will fuse and they'll give the almighty zygote. Almighty zygote, students. Right. This zygote is always diploid in nature because gametes are fusing, students. Gametes are fusing. Zygote is diploid, students. Zygote is diploid. Okay. Okay. Done, students. Now, this zygote will now undergo nice division and it will give you one diploid structure. This particular zygote, student, this zygote can undergo a process and it can give you a structure called a sporophyte, which is again diploid, students. Diploid. Now, students, here do I write mitosis or meiosis again? All of you, do I write mitosis or meiosis again here? Again, mitosis. Equational division, not reduction. Equational division. Okay, mitosis again. Because ploid is maintained, no? Ploid is maintained. Ploid is always maintained here. Okay, ploid is always maintained here because haploid, diploid to diploid only. Yes, students. Now this structure here, sporophyte, students. Sporophyte will produce what? Sporophyte will produce spores. Now, students, these spores are haploid in nature. Tell me, students, what will happen here? Differentiation, students, differentiation. See, zygote is one cell. Zygote is one cell. Sporophyte is multicellular, right? How can we go from one cell to many cells? We need to do mitosis, no? No, students. Now, here we have meiosis. Okay, meiosis, students, meiosis. Now, these spores will develop into the main plant body by mitosis again. Students, but, what is the but here? Haploid only. Students, the but part here is that in the case of algae, what happens, students? In the case of algae, this may be true for other, right? In the case of what happens, algae, sporophyte stage is not present generally. These spores directly develop into, this zygote directly develops into, students. This particular zygote directly develops into spores here. Directly develop into spores, zygote. Now this process is called as what? Zygotic meiosis. That is, meiosis occur inside zygote. Meiosis we have zygotic meiosis, students. Zygotic meiosis happening here. Zygotic meiosis. Done. Students, usually what happens is that if the differentiation has to happen, Tamil, right? If the differentiation has to happen, right, it needs to undergo mitosis eventually because it needs to increase in cells, right? It needs to increase in cells. So we need mitosis. A zygote can just become differentiation just bigger. Differentiation is just becoming bigger, right? You can't just go directly, right? Crazy, no, students? Crazy. That's also the life cycle. Plant body, main plant body, independent. Sporophyte is not present. Zygotic meiosis. And students, last thing after live is economic importance. Last thing is called as economic importance. Now, economic importance, students, there is a meme which I always like. I'll share the meme in my Telegram also, students. I'll share the meme in my Telegram also. That majority of your oxygen comes from where? Everyone says it comes from where? The trees. But majority of your oxygen comes from the from where? From the algae. Majority of your oxygen comes from the algae generally, because algae are present in aquatic condition. Algae are present in the aquatic condition, and aquatic condition, lot of algae, more water, so more oxygen, students, more oxygen, right? More number of oxygen can be there, right? Now, apart from that, in the case of aquatic ecosystem, algae has to produce food, so it is present in the first trophic level everywhere. It has to prepare food, first trophic level. Yes, first trophic level, students, right? First, one second, students, one second. Some eyes are hurting suddenly because there are a lot of lights in the room, right? One second, students, one second. Okay, students, done. Right. There are a lot of algae usually becomes the first trophic. It is a part of the first trophic level everywhere. First trophic level, right? Now, students, now, apart from being the first trophic level, as well as being part of your food chain, the first trophic level, and producing a lot of oxygen, they also produce other substances which we use. We are greedy, always, right? There is something called as Gracilaria and Gelidium. Gracilaria and Gelidium, right? They are used to make jellies, they're used to make ice creams, they're used to make agar-agar, right? Agar-agar. Now, agar-agar is used in your gel electrophoresis. It is also used in your microbiology to make the medium also, where we grow bacteria, right? The medium that is made using agar-agar here, jelly and ice cream. Sargassum laminaria profile are used as food also. Chlorella is your space food. Certain students, I don't even remember NEET score. It was 10 years back. I don't remember. Right. Certain marine brown and red algae produce large amount of your hydrocolloid, that is your carrageenan and algin. Carrageenan and algin can also be used. Okay, algin can be used. So all these are economic importance, students. Students, PYQs I'll upload in my Telegram right now. Students, PYQs I'll upload in Telegram. Yes or no? Let's just see what have we covered till now, students. Students, I wrote NEET examination in students, all of you might know that I was born in Ayodhya, right? I have a domicile certificate of UP, right? UP. That's why my Hindi is good, right? And I was in Gujarat also for a couple of years, so I have a domicile from there, starting years. Then I came to Karnataka. So when I wrote my NEET examination, I don't remember, but I got a seat. I could have gotten a seat in the UP, right? But my entire family was here. Like, how do I travel from here to there? It was not possible. And leaving the entire family from here to there would have been the most difficult task of my life, right? Staying alone in different state altogether because of the different domicile, right? So I wrote NEET examination in 2016. I wrote 16, I guess. It's going to be almost a decade now. Almost a decade in 2016, right? And between, it was the most difficult paper ever. Second most difficult, I guess, now. Now, students, let's go. Okay. Right. Now, students. See, did we cover all this? Students, did we cover all this information? Let's just see. We know that all of you students, if you're able to understand the line of NCERT, fire emoji in the chat. Very simple rule. Okay, very simple rule, students. Okay. We know, students, that, buckle up all of you, buckle up for the fast track. All of you, we know that 5 kingdom classification was given by R.H. Whitaker here. We have the Monera, Protista, Fungi, Animalia, Plantae. Done. Now, students, we are talking about plant kingdom right now. Now, students, initially what happens? We must stress here that our understanding of plant kingdom has changed over the time. Fungus, a member of Monera and Protista, having cell walls, have now been excluded from Plantae. So students, initially what happens? Initially, all of you, students, initially what happened? Initially your fungus was here. I told you students, initially I told you fungus was present. Fungus and Monera were part of your this thing itself. Then they were excluded. They were excluded separately. They were excluded separately out. Okay. Now, right. So cyanobacteria, that are also referred to as blue-green algae, is not algae. All of you know that. Cyanobacteria is not an algae. Cyanobacteria is technically part of your Monera. All of you know that, students. Now, we talk about artificial system of classification given by Carl Linnaeus. Artificial system of classification is based on morphological features alone. Then natural system of classification was based on what? The internal features. Okay, right? Internal features like your anatomy, structure, embryology, phytochemistry. It was given by Bentham and Hooker classification. Then students, we have phylogenetic system of classification was given by whom, students? Phylogenetic was given by the Charles Darwin here. Okay. Now, whenever we have fossils, we can use fossils and we can do phylogenetic system of classification. If there is no fossil, we go for alternatives. Such as your numerical taxonomy, students. Read the lines. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characters, numbers and codes. Remember 1010, I told you. Numbers and codes are assigned to all characters and data and then processed. This way each character is given equal importance and same time hundreds of characters are considered. Cytotaxonomy based on chromosome number, chromosome behavior. Yes, right? Chromosome number, chromosome behavior, structure. Then we have chemotaxonomy based on chemicals. Then we talk about algae, students. Algae is always chlorophyll bearing, simple, thalloid, autotrophic in nature. I told you. Students, what is the meaning of thalloid? The meaning of thalloid is that they do not have true stem, true root, true leaf. Nothing is present. Nothing. Undifferentiated. Students, undifferentiated. Not students. What is the habitat? Fresh water and marine, but they can also be found on moist rocks, soil, wood, everywhere. They are also present in the symbiotic association with fungus in the case of lichen, or even in the case of sloth bear on the top body of it, students. Top body of it. Okay. Now, students, they come in different shape and sizes. Yes or no? Like all of us, right? They can be colonial, volvox, filamentous, Eurothrix and Spirogyra, or they can be hundreds of meter tall, like giant kelps. Giant kelps are massive plant, 100 meter tall, 100 meter tall. Okay. Now, students, algae reproduce by vegetative, asexual, sexual. Vegetative by binary fission, fragmentation. Asexual, students, by zoospore formation. See, students, here. See, vegetative by fragmentation, each fragment. Yes, zoospore is where asexual type is zoospore formation. Sexual, we have three types: iso and iso, as well as oogamy. Examples. Friends, can you see here? Sexual reproduction through fusion of two gametes. These gametes can be flagellated and similar in size, right? As in Eurothrix. Eurothrix is motile. Non-flagellated. Non-motile is your Spirogyra. Isogamy. Anisogamy, Eudorina. Then we have your Fucus and volvox is oogamy. Fucus and volvox is oogamy, students. In oogamy, what happens? In the case of oogamy, one is large, one is tiny. Tiny is motile, large is non-motile. Now, students, here are different shapes. Students, here if you see the brown algae has root-like structure called as holdfast, stem-like structure called as stipe, leaf-like structure called as frond. Holdfast, stipe, and frond. Please, students, remember that. See, students, this is your porphyra, the red tissue paper. Porphyra, the red tissue paper here. Red tissue paper here. Now, students, these are the economic importance. This you can read on your own. Economic importance are given here. Now, students, chlorophyce. Understand, chlorophyce. Chlorophyce are commonly called as green algae. All of us know that. Unicellular, chlamydomonas, colonial, that is volvox, filamentous, Spirogyra. Okay. Now, students, chloroplast may be different shape. I told you different shapes of chloroplast. Discoid, plate-like, reticulate, cup-shaped, spiral, ribbon-shaped. Done, students, done. Now, most of the members have one or more storage bodies called as pyrenoids. Look at the chloroplast. Pyrenoids contains proteins besides starch. So pyrenoids help in protein storage as well as starch, students, starch storage. Okay. Now students. Now cell wall, inner layer cellulose, outer layer pectose. Yes, sir. Now asexual reproduction is by flagellated zoospore formation. All of you know that chlamydomonas zoospore formation happens. Yes or no? Now students, the sexual reproduction is shown by considerable variations here, right? Isogamy, anisogamy, oogamy. Commonly found in green algae are your, all three are present in green algae. Examples: Volvox, Urothrix, Pyrogera, Cara. Now, students, brown algae. Brown algae, students, primarily found in marine habitat. Sargassum was the example there. Yes or no? Sargassum was the example there. They show great variation in size and form. Giant kelps are there, right? Ectocarpus. Remember, I showed you the photo of Ectocarpus, students. This photo of Ectocarpus. Remember Ectocarpus story photo I showed you? And profusely, students, Ectocarpus was profusely branched. Yes or no? If you go back here in my image I showed you, Ectocarpus. Students, see the Ectocarpus. See the branching Ectocarpus. Profusely branched, right? Profusely branched. Profusely branched Ectocarpus. Profusely branched forms. Right? Two profusely branched. Even Ectocarpus filamentous, and giant kelp is also very large. Very large. Heights of 100 meters. Chlorophyll A, chlorophyll C, carotenoids, xanthophyll. Now students, this xanthophyll called as what? Xanthophyll called as fucoxanthin. Fucoxanthin can vary from high concentration brown, low concentration olive green. Xanthophyll pigment. Now students, here. Food stored as complex carbohydrates. Mannitol and laminarin storage material. Algin is the hydrochloride substance. Outside by gelatinous coating called as algin. The protoplast contains additional plastids, a central located vacuole, holdfast, stipe, and frond are the three different parts there. Okay. Now, vegetable reproduction by fragmentation, same thing. Asexual by your brown reproduction, most brown algae is by bioflagellated zoospores, same thing. Same thing. See here these bioflagellated zoospores that are pear-shaped have two unequal lateral attached flagella. Students, didn't I tell that? Two unequal. Chlorophyce, did I miss that line? Chlorophyce, did I miss that line, students? It's not mentioned anymore. Apical equal. Okay. Now, students. Now you can see, students, here. Sexual reproduction is by isogamous, anisogamous, oogamous. Union of gametes may take place in water or within the oogonium here, oogonium species. The gametes are pyriform again. The gametes are pyriform, pear-shaped and bear two lateral attached flagella, one long, one short. Okay. Examples: Ectocarpus, Dictyota, Laminar, Sargassum, Fucus. Red algae, ancient algae, pigment are phycoerythrin. Yes. Mostly are marine, present in your light regions, well-lit regions here. Yes. Red thala is the most red algae are multicellular. Most of them have complex body organization. The food is stored as Freudian starch, similar structure to amylopectin and glycogen. Red algae usually show fragmentation. All sexual by non-motile spores, non-motile gametes. Students, here it is seen. 2 to 8 equal apical. Okay. Sexual reproduction is oogamous and accompanied by complex post-fertilization development is seen in the case of your red algae. Polysiphonia, the beautiful one. Porphyra, the red paper, tissue paper. Grasslea, Gelidium are the jelly making factory. Okay. Students, with that, chapter is over. Chapter is not over, algae is over. And introduction is over, students. Introduction is over. Okay. Okay, all of you? Everyone? Done? Students, today I will share the DPPs today only. You can check my Telegram. I will share the DPPs now only. I'll share after I eat my dinner. Right? Because this eye is hurting a lot right now. I can't see the light next level. It's hurting. So I will make you solve all the questions later on, students. If you want, when I'm teaching you Bravo 5, that time I'll teach you. Apex is tip only, right? Apex is same thing. Tip and apex is the same thing. This is the tip, apex. Tip and apex is the same thing, students. Okay. Okay, students, bye all of you. Let me know in the comment section how was the class. Students, all I can see a lot of them are from DNA, some of them are non-DNA. Right. So students, I have thought in DNA batch I go more doubt solving process. Right. So Gujarati I can understand. Right. In DNA batch, that's a two-way conversation. Here it is one way only. It's mainly one way right now. In DNA, I always make it two-way conversation. Two-way conversation in DNA batch. Right. So if you're not a part of DNA batch, students join. Right. I'll be starting, students. I'll be starting anatomy of flowering plants from the coming week. So if you can join DNA batch, you will be starting from anatomy with me. So you will love it, anatomy. Okay, students. So bye bye all of you. Take care, students. DNA batch, all of you know. Link in the description. You can go join from there. So good night, students. Bye bye all of you. Take care. Notes will be given in Telegram channel. You can download from there. And bye bye all of you. Bye bye, students. Bye bye.